Thanks, Akshay. Thank you very much for your welcome. So, as Akshay says, I'm a nanotechnologist, so I lead a large research group in the Cavendish Laboratory, which is the physics department of the University of Cambridge. And my team and I, we spend our days looking at the near atomic scale. That's what nanotechnology is all about. So what I'm going to do this afternoon is give you a very brief overview of this new world of nanotechnology. So I'll tell you what it is, and I will tell you what's exciting about it. And then I really want to shock you, because it's hard to shock people these days, but as a scientist, I want to spend 2 minutes at the very end talking about God. So there we go. Nanotech, what is this? What is God? Okay, so this is nanotechnology, and this is a size scale, so you all know what a meter is, that's with me. If we go down a bit smaller to a millimeter, you still know what a millimeter is. If we go down another factor of 100 from there, we're now getting into a realm where it's a little bit difficult for us to imagine what that corresponds to. But the picture you're seeing there is a pinhead with a human hair and a little dust mite on it. And those things come in at around 50 millionths of a meter. If we go down a little bit smaller to about 10 microns, so 10 millionths of a meter, we get to things like single red blood cells. And then if you go all the way to the bottom of that scale, down to a single atom, that would come in at about 0.1 of a nanometer, or a 10 billionth of a meter. So nanotechnology is everything that lies in that gap. So it's everything that's from roughly a millionth of a meter down to a billionth of a meter in size. And just to give you an idea of the sorts of things we can play with in that size gap, that's an abacus. I'm sure you know what an abacus is. You probably haven't seen one like that before, though, because instead of having wooden legs, that abacus is made from single molecules. And it was actually by IBM quite a few years ago now. They showed how you could actually count with it. They moved the molecules around, and they spelled out the numbers 0 to 10 just by where they put the individual molecules on the surface. So how do we do this? Well, there are some key techniques, and I'll show you those now. You're then all good to go and set to do your own nanotechnology. And one of the most important techniques is called atomic force microscopy. And it's no more complex, really, than a blind person reading Braille. But instead of using human fingers, it uses super sharp, atomically sharp fingers. So what we do is we make a very pointed tip, something that's sharp to within about 10 atoms or so, and we connect it to what's called a cantilever, which is really just a diving board. And as that very sharp tip passes over the surface, it goes up and down with the individual atoms as it passes. And we bounce a laser beam off the back, and by looking at how the laser beam is deflected, we can actually see the individual atoms. There's a sister technique called scanning tunneling microscopy, which is very similar, but it's only using a laser beam and measures electrical current. But with a combination of those two, you've got a microscope where you can see things atom by atom. That's pretty amazing. I don't know if you're amazed by that, but if we'd been having this conversation 100 years ago, we would have been discussing philosophically whether material actually was made up of atoms or whether it was all one continuum. Now we can just take a piece of whatever, put it in the microscope, and you can see the atoms directly. One of the ironies of nanotechnology is the smaller you want to make things, the bigger the machines you need to build. So my lab is full of huge lumps of stainless steel like this. Everything has to be done in a very high vacuum of the same level as outer space. Because if you have a passing air molecule that collides with something so small, it'll destroy it. So we need to work in an environment where no air molecules collide with the surface for many minutes at a time. And that really needs very high quality pumps and all sorts of vacuum. So let me, those are the basic techniques. Let me introduce you some of the basic materials of nanotechnology. And surprisingly, carbon is actually a really important material in nanotechnology. When I was your age, I was taught that carbon comes in different allotropes. There's graphite, there's diamond, and we've now added to that list 3 other allotropes, all of which are nano-enabled. So there's what's called C60, where if you imagine a football, imagine the hexagons and pentagons that make up a football. If you put a carbon atom at each of the corners and link them all together, you get 60 atoms in one little football molecule. And that was actually discovered in space. So Harry Proto was pointing his telescope out of space and was looking at the different chemicals he could see, and he saw a signature of what he could only explain if it was clusters of 60 carbon atoms together. Now we synthesize these things in the lab on a regular basis. There's a different nano form of carbon called carbon nanotubes, and if you imagine chicken wire, imagine the stuff you make fences from, and you roll that up into a tube, that's pretty much where the atoms lie. And then you can actually think about unrolling that, so you've now got a single sheet of carbon atoms connected together hexagonally, and that's called graphene. And that also won the Nobel Prize just 6 years ago. The guys in Manchester won the Nobel Prize. So why do we bother? What's the reason we should spend so much time on this particular size scale? And it's really what we call the nano advantage. The nano advantage is that basically the laws of physics are a little bit different in such very small things. So the properties of materials change in quite a dramatic way if you make things that small. And quite often that changes for the better, and we can potentially use those new properties to build better, cheaper, cleaner, smarter materials, products, gizmos, whatever you like, in a way that wouldn't be possible if we were using the normal properties that nature gives us on a larger scale. So this is really important. I don't want you to go away thinking that this is just all about miniaturization, because that's not really interesting. Just making the same thing smaller isn't particularly clever. What's really smart about nanotech is that it allows us to access new properties that have no corresponding equivalent on a larger scale. Let me give you just some examples. So if we were back to 1947, that's what a transistor would look like. You'll know transistors are what make up the hard microchips that make the modern world modern. So that thing there is about that big. This is a big single transistor. Scroll forward to 21st century, and that is what a transistor looks like there. It's pretty much the same basic principle, but instead of being that big, the thing is 65 nanometers across. So just a few hundred atoms in size. As a consequence of that, we can make wafers of these things with hundreds of chips on, and each chip containing many hundreds of millions of transistors. I'm sure most of you have got smartphones. The microprocessor that drives your smartphone has in excess of 100 million transistors on it. And it's why the phone can do all the things that it can do in a way that supercomputers would be needed even just a few years ago. Healthcare is another area where nano is really making a difference. This is some work from a friend of mine at the London Centre for Nanotechnology, just down the road in South Kensington, in Bloomsbury. And she's investigating why it is that some bugs, things you catch if you go to hospital, these immune MRSA, things like that, the ones that don't respond to antibiotics, as to why they are so good at avoiding the things that normally kill bugs. And she did a very smart experiment. I'm not sure how clearly you can see that, but she took the cantilever of the atomic force microscope. Rather than having using the tip that's usually on the end of it, she used the cantilever as a really sensitive thermometer. And she set up molecular reactions between the bugs and antibiotics on the back of the cantilever. And by measuring the very small amounts of heat evolved during the reaction, the cantilever bends one way or the other. It's a bit like the switch that controls your central heating in a house. It's a bimetallic strip, and the hotter it gets, the more it bends one way, and that switches on the heating. Because we're using our laser, we can measure movements of this cantilever with subatomic resolution. We can consequently measure tiny, tiny amounts of heat and allow us to work on pretty much single molecules with each other. And she discovered that it's actually one extra proton on the side of the bug that allows it to avoid antibiotics and what gives it its antibiotic resistance. Renewable energy is another really important area, because one of the problems in renewable energy is that we don't always have the materials that we want to make the sorts of new technologies that we need to reduce carbon footprint. And solar cells are a great example, photovoltaics. There are many materials out there that actually convert light into electrical energy particularly efficiently. And it's a real shame, because if you work out how much energy lands in each square metre of even a place like England, it's somewhere just short of 1,000 watts per square metre. And actually that is plenty. You compare that to how much energy we actually consume. If we could really extract all of the energy landing from the sun, we would have no need of anything else. The problem is how to convert it with a reasonable efficiency. So the nano solution to this is, if the materials to do that conversion don't exist naturally, let's make some new ones. Let's create some new materials with new properties. And what we do is we stretch and we squash atoms in order to make them behave in a different way, to make them better at capturing sunlight. So we start with three materials that are all in the same family. We start with gallium arsenide, which is a very important material. And then we added a phosphorus to make gallium arsenide phosphate. And we added a bit of indium to make indium gallium arsenide. Now the spacing between the atoms in those three materials are slightly different. So you see on the left of the screen there, you've got the gallium arsenide at the bottom. The gallium arsenide phosphide is a little bit smaller, the atoms are a little bit closer together, and the indium gallium arsenide, the atoms are a little bit further apart. We basically then glue those together. Atom by atom, we connect them into what's called a heterostructure. And as a result, all the atoms have to adopt the same spacing. So the gallium arsenide phosphide has to stretch out a bit, the indium gallium arsenide has to squash in a little bit. And the remarkable thing about electrons is that if you squash them or you pull them out, you change their properties dramatically. And in doing that, we create a new material that is really efficient at capturing sunlight and converting it almost without loss into electrical energy. So that's another place it's being used. It's great to show fun toys and nice technology, but one of the things we as scientists need to get better at early on is asking about the ethical implications of our technologies. For too long, scientists have been pressed on making their new discoveries and to let other people worry about whether it's a good thing to be doing as a community. And nanotechnology is one of those areas where we really have to get this right in advance rather than be behind the curve and worry about it later. I think there are four things that we need to consider in trying to weigh up if nanotechnology is an ethically good thing to be doing. We need to look at the safety. We don't want to be poisoning people. We need to balance the safety against global challenges. So things like renewable energy, carbon footprint, global warming, aging populations, lack of water, all these big societal challenges that are going to be huge problems for all of your lifetime. We need to work out what benefit can be brought to those huge questions by the technology. We need to look at the proportionality of the risk. We need to make sure that if something goes wrong, it doesn't wipe out the entire planet. And we need to look at nature and decide if what we're doing is being done already in nature, or whether this is actually the first time on the planet this has been done. I'm not quite sure how we interpret the answers to that, but it's always good to know if we're going in places that have never been used before, or if actually we're just repeating what nature already done. So let's just look quickly at some of those. So in terms of safety, this is the way we think about it. We look at the amount of material on the bottom axis. Are we talking about bucket loads, or are we talking about a few atoms amount? And we look at the accessibility of the material. In other words, is it floating around in your blood, or is it struck down firmly to a surface? So how much of it is there, and how easy is it for it to get into you in a way that can do some harm? And depending on where you are on those two axes, determines how risky we would see this as being. So for example, if you are up here in the exclamation mark area, that means you've got lots of nanomaterial and it's everywhere inside you. And down at this end, it means there's not actually that much stuff and you can't get at it anyway. So the microchip, or your mobile phone chip, is down there. So you've all got a nano machine in your pocket already, but there's nothing to worry about because there's only a few nanograms of actual transistors in there and they're all thermally attached to the substrate. If you contract a nasty disease and you have to have an MRI scan, in coming years we might use nanoparticles to try to identify the tumour or whatever it is that we're looking for. So in a case like that, you're not fully exposed to the material, it will give you a drink to consume, but actually it's still just nanograms, and so it's not that much. One of the areas that people are looking at is sunscreen, trying to make things like titanium dioxide nanoparticles, which absorb ultraviolet light very efficiently. And using those is a nice way of protecting your skin from the sun's harmful rays. And that's an interesting one, because there you've actually got relatively large amounts of it. You slosh it on, and you're putting it in direct contact with your skin. So you need to be quite careful there. And so that's the general methodology we use to try to decide how safe these things are. Let's just look briefly at this question of, is this new to nature, or has it been done before? Let me introduce you to this chap, the morpho butterfly. I don't think he flies around Windsor, but certainly quite common in South America. And it's this beautiful iridescent blue colour in the wings. And what's really interesting about the morpho butterfly is there's no pigment in the wings. It doesn't get its colour by a pigment or a dye. It actually gets its colour by natural nanostructures. If you zoom in on part of the wing and image it in the very high magnification, what you'll actually see is row after row of little naturally occurring nanostructures. And this is called structural colour. And it's, I guess, a very simple form is the hologram on a credit card or something like that. You know, if you tilt that in the light, you see different colours. That's structural colour. And it seems that nature has learned how to use this technique far more interestingly than credit card holograms. And the morpho butterfly gets all of its colour just by natural nanostructuring of its wings. We don't actually have to go as far as South America to find this. This room will do. So the thing that keeps us all alive, the engine of our metabolism, is an enzyme called ATP synthase. And this is what makes the rocket fuel inside you, which drives everything about you. It drives your muscles, drives your metabolism, gives you life, basically. And if you look at how ATP synthase works, it's actually a little nanomotor. So you've got one set of molecules that act as the stator, that's the part of the motor that's fixed and doesn't move. And then you've got another part of the molecules that are inside the stator called the rotor, which actually turn. And within this little molecule, the center part is rotating, and as it goes, it strips off protons and bangs them together. And it's really like an old Victorian-style machine. Imagine Wallace and Gromit. Imagine one of those amazing contraptions. And it's doing this on the atomic scale, taking single protons, bashing them into other molecules, and making the very stuff of life. From an evolutionary perspective, this is actually very old. It's been powered by ATP synthase for a very, very long time. So actually nanotechnology has been nature's chosen way of powering life pretty much since the very beginning of life itself. Okay, I want to shock you, so I just want to talk just very briefly, just a little bit before we take some questions, about theology. And part of the reason I want to talk about this now is it's directly relevant to nanotechnology, but also I just want to take a little time to counter the idea that all scientists are atheists. But it's not true. There are a number of us who have religious belief, and we try to think about our work in terms of that. And there's a really interesting little role that nanotechnology is playing in these bigger questions of Big Bang and evolution and that sort of thing. So at the moment, science has a fairly good picture of how everything came together, starting with a Big Bang, which then creates individual protons initially, and then ultimately atoms, and then ultimately molecules. And then those molecules come together and form bigger molecules like DNA and RNA, these complex molecules that you need for life. And then ultimately those big macromolecules can form living organisms like single cells. And once you've got single cells, you're then into animals, plants, people, you. So this is the general sort of chain that's been pieced together. If you look at the detail though, it's not all quite there. And specifically, the bit that gets you from the Big Bang to individual protons and then atoms, that's great. The bit that gets you from small atoms to small molecules, that's all really well understood. But to be honest, the bit that gets you from small molecules into complex molecules for life, like DNA and RNA, the science is a bit shaky. There's really not much what I would consider to be hard scientific evidence for that particular step. And likewise, the bit that then gets us into those very first cells, we don't really know how that works. But once we got the first cells, we've then got a pretty good idea how they can then expand to become complex beings like you and me, animals, plants, and people. What's really interesting is that range, going from single molecules to more complex structures, is absolutely the territory of nanotechnology. This is where nature's nanotech was fully at work. And so I can't tell you what the answer is today, but I would predict that in the coming decade or so, we're going to get some quite remarkable scientific breakthroughs that allow us to complete that chain of life and allow us to have a decent scientific understanding all the way from the Big Bang, all the way through molecules, cells, and ultimately people. And the question that I want to sort of throw out now and have you think about in advance of this is, if we complete that chain of understanding, does that fundamentally change anything theologically? Do we, having understood the scientific mechanism, do we then stop talking about God and things like that? Personally, I would argue we don't. I think that's actually a theological mistake called God of the Gaps, which says that as science progresses, the need for God recedes, and I think we should avoid making that. But it's good to think in advance, because there's going to be a temptation to think these things very soon once we fill in those missing links in the nano regime. So that was pretty much what I wanted to show you. So briefly, nanotech, it's the art and the science of manipulating material on this nanometer scale, and it's on track to form the next industrial revolution. If handled correctly, it has the potential to be a significant force for good, and not just in smarter phones, but in things that really change the world. Things like global warming, access to water, new antibiotics. Actually, God got there first. Nature is full of nanotechnology. The very stuff of life is powered by nanotechnology. And my view is that in the future, this isn't actually going to change the relationship between God and science. And that is all I have to say. Thank you very much for listening. Very happy to take questions.
So looking at how much progress we've witnessed in this field in the last 20, 30 years, do you think that's going to be a similar amount of progress in the next 20, 30 years, or do you think it's going to magnify?
Interesting question. So one of the phrases that's sometimes used in science is standing on giants' shoulders. With Isaac Newton, who said, "I've seen further than other men, it's because I was standing on giants' shoulders." So essentially it means this generation of scientists can start from where the last generation left off. And so, using that principle, maybe we will see more rapidly accelerating progress, because you're starting with a number of things already in place. I think there are a few caveats. One of the industries that I'm particularly interested in is the hard disk industry, for example, storing data. And on the one hand, that's very much a nanotechnology business. And the cloud, for example, has enormous requirements for data storage. And yet, if you look at what's happening in the research community, people are pulling out of research in that subject because they think we've basically reached the limit, and that they don't think there's too much more progress to be made. Now it's just a question of the industries making the same stuff bigger and cheaper. So progress tends to go in spurts, and then it tends to fall off a bit when people reach usually some sort of economically imposed limit. It's just too expensive to go any further in this field, and the attention then shifts to somewhere else. So as to whether the progress will always be in nanotechnology, I really can't say. Maybe we decide that it's not worth the effort to go past a certain point, but I'm sure there'll be other good things coming along.
Hi. When these nanoparticles, when using them, how would you supply energy to them so that they can move, and also how would you transmit signals to control them?
Yeah, that's a good question. So how do we move nanoparticles? How do we transmit signals to them? We've got lots of ways. My own interest is using magnetic fields. So we make the nanoparticles out of magnetic material, and in some work I'm involved with at the moment, we put them into cancer cells. So we can use biological markers that make them attract into just cancer cells. And then once they're in there, we apply a magnetic field to the entire patient. And at that point, they start spinning. And they become like little nanoblender, and they mash the inside of the cancer cell and kill the tumour. So magnetic fields are a very good way of doing that, because the fields are otherwise harmless to the human body. You can use infrared light. So infrared light passes through bodies and lots of things, and the particles often absorb that light and get very hot, and you can use the heat to, for example, bend something, burst something, so you can communicate with the particle. It was interesting you said communicate with them. I think the old model that we think of miniaturized technology is that it's basically the same as bigger technology but shrunk smaller. So it was a wonderful, I think it was 1960s movie, sort of sci-fi thing, Fantastic Voyage, it was called. The idea was that, again, there was a patient with cancer, and some scientists got into a little submarine, and the submarine shrunk down to the nanoscale, and then navigated its way through the body until it found the tumour. That's not the way we think about these things now. We don't assume that the smaller version will have the same properties as the larger version. So maybe we don't need to send information to them. Maybe we don't have to communicate in the way we communicate across a radio channel to a larger thing. It may be that we can, for example, get information back by a collective process where no single particle is communicating, but the whole ensemble of them together indicates information. So different ways of thinking about communication.
Do you think that after you've used nanotechnology for first aid and things like that, do you think that you could actually make them so that they could sort out cells? So for things that where you could, for babies in the womb, you could make them so that they had certain genes that would make them better to almost make us evolve into better.
Genetic manipulation.
Yes.
There's an ethically hot subject. There are probably, I think yes, I think there are things that can be done there. So whether we want to do them, that's also an important question. I think in that particular branch of science, the molecular biologists have probably got more tools at their disposal than we have. Molecular biologists have really got amazing ranges of molecules that can do all sorts of things. Where we are thinking about sending in the relatively brute force thing of nanoparticles, where molecular biology has completely run out of options. And cancer therapy is one of them. I think we will do something, you mentioned sorting. One of the things we can do is potentially sort cells. So there's a technique called flow cytometry, which is used in hospitals at the moment, where people take samples of the blood and then they break it into individual cells and they count them as they go by, and they count all the different types. And sometimes you can imagine then sorting those so that if it sees a leukemia cell, for example, it spits it out, whereas lets all the healthy cells go through, and then you, almost like a dialysis, you then put clean blood back into the patient. I think we could definitely imagine those sorts of technologies.
There was there was a film I once watched called The Island, which is where there's like clones, and I was wondering that not specifically clones, but if you had like separate like cells in which you could use if there was ever a problem, in which you could use if there was ever a population problem that could help the human race if there was ever some kind of extinction problem or something.
Yeah, maybe even earlier than extinction, maybe treating diseases. So malaria, for example, there's one of the big problems with malaria is that if you get bitten by a mosquito, you can carry one phase of the disease without actually showing any symptoms, so you remain healthy. But if another mosquito bites you, you infect the mosquito, and that's one of the important ways malaria was passed. So it's actually not about keeping mosquitoes healthy, not about keeping the humans healthy. And I have a colleague in Australia who's working on a nano-enabled technology for that to identify people who are carrying a harmless form of malaria, but just to make sure that they stay away from places where they can pass it back to mosquitoes.